Hello, welcome to your student selected module in research. My name's Dr. Anna Collins and I have been asked by Mr. Abdul to deliver this talk to you on research and medicine. So my background is that I've completed a PhD after a 3-year clinical research fellowship, and I hope that I'm able to inspire some of you to undertake a period of research during your higher medical training. So today I'm going to talk to you about the types of medical research, research governance and ethics, and career pathways if you wish to pursue an academic career. So how do we classify medical research? A really simple way to do that is to split medical research into primary and secondary surveys. So from a primary point of view, they're the studies, the actual research work itself, and this can be subcategorized into basic or experimental research, and this deals with understanding physiology, drug mechanism of action, how the body works, and trying to gain insights about the basic medical sciences behind clinical research. Then you have clinical research. These are either experimental or observational studies, which look at how we influence patient care using clinical research. And then on a higher level, you have epidemiological research, which focuses on population health. Now, secondary research is more synthesis of existing research. So this could take the form of a narrative literature review or a systematic review or meta-analysis. So let's have a think about experimental research. And the reason I've chosen to use this picture of a jigsaw puzzle is because when you are conducting experimental research, it's likely that you'll use a number of these different types of research studies to prove your principle or prove your hypothesis. So, for example, in drug development studies, you may use cell lines or cell studies to test new agents. You may want to do some genetic studies to understand the tumour biology in a little bit more detail. You may use animals, so for example mouse models, to test drugs by giving drugs to mice or by growing tumours in mice, so that you're therefore able to understand tumour biology a little bit better, avoiding the use of human subjects. And then you've got omics. Omics refers to a range of different disciplines, so genomics, proteomics, metabolomics. And what this means is looking, although we have different genetic processes that we understand, how far do these genetic processes translate into what happens in the cell? So which proteins are synthesized and how does that affect later metabolomic processes in cellular function? So it's trying to look at the end effects of genetic changes. Analytical studies are studies where you optimize work in order to support your other work. So things like optimizing enzymes, optimizing antibodies for use in multiplexed immunohistochemistry. And it's really useful to do some experimental research at some point in time in your clinical career if you're considering a clinical academic career, because it really gives you the grounding in how we develop proof of principle and how we develop concepts. And it's very relevant to clinical work. So moving on to clinical studies. So you can generally categorize these in analytical or descriptive studies. When we first look at analytical studies, these may be experimental or observational. When you look at the experimental arm, this involves giving an intervention to a patient, such as in a randomized control trial or a crossover study, and having a look at the effects of that study. In an observational trial, this doesn't involve giving any interventions, but it does involve looking at cohorts or groups of patients over a period of time. A cohort study is looking at people exposed to a certain variable and seeing what happens after that exposure, whereas a case control study is usually retrospective, so it's taking people who have a disease and having a look back and seeing what exposures could have been responsible for that disease. In terms of descriptive studies, you have surveys. These could take the form of patient surveys, professional surveys, or Delphi exercises. And the Delphi methodology is a way of synthesizing expert group opinion. And what happens is you have different rounds where experts are asked to look at statements and evaluate how they feel about them to try to achieve consensus on a group or a spectrum of ideas. And what you're looking for is to achieve a point where a group of experts all agree on something through a standardized methodology. And that might take the form of initial surveys or working round group tables. Then you have qualitative work. Qualitative work may take the form of semi-structured interviews. And what happens is researchers try to draw out themes from talking to different participants to try and find common themes from their descriptions of their experiences to bring out in the research. So you want to conduct a piece of research. The first thing you need to establish is what is your research question? Now, when you're designing a research question, you need to have a very specific question to test a specific hypothesis. So first of all, you're going to take the topic that you're interested in and conduct a literature review. You then want to narrow that topic down and focus it onto the specific question that you want to answer. And ideally, your research question should be original. It needs to contribute something new to the literature. It shouldn't be a repeat of what is already known. So you need to think of a new angle. It might be that you're doing something that's already done, but in a different cohort of patients. It might be that you take a new angle on a piece of work which hadn't been looked at in that way before. It should be relevant and important to patients. And one of the most important things is that it's feasible so that we can actually achieve it. And that's where trying to have a robust methodology and a really strong trial design to answer your research question comes in. You also need to be pragmatic. So it can be very difficult to do research on very rare conditions. And that may mean that some of the higher quality research trials are not practical to do in rare diseases. And this may change your research methodology. You also need to think about costs, about personnel that you need involved, and whether you have the funding to be able to support this. And then research should be ethical. So you need to consider the impact of your research on participants and whether that is appropriate. So one way to think about designing or formulating a research question is to use the PICO framework. So this means that your research question should have the same structure. So first of all, P, patients. So who are your participants going to be? What is the problem? It might help you to think of this in terms of disease, condition, severity stage, demographics. Be precise about who you want to do the research on. Intervention. So what's the proposed intervention or the exposure? So for example, for a drug, you may need to specify dose, duration, timing, the route of administration. What is your comparison group? Is that standard of care, no treatment, placebo? You should have defined that in your research question. And finally, outcome. What's your endpoint? So this could be anything from quality of life, survival, morbidity or mortality. So your research question might start something like, is it useful to start aspirin at 12 weeks in women at risk of preeclampsia? But that's not a very specific question. So how could we make that better? Why don't you consider, does starting aspirin in women at risk of preeclampsia at 12 weeks gestation reduce the risk of preterm delivery compared to standards of care, no aspirin? That's a lot more specific and a lot easier to design a research protocol around. So why don't you have a go at coming up with some PICO style research questions? So once you have your research question, you may want to develop a research proposal. So how are you going to go about doing your research? Typically, this will involve the title, background, so establishing the context and the rationale for your research, and the findings of your literature review, your PICO research question, and then your proposed research design and your methodology. And I'm going to go on and talk to you about the different types of clinical trials in a moment. You should touch on the implications and the contribution to knowledge of why your research is important, the finance you're going to need and your plan for the allocation of this finance, the time plan, and when you plan to achieve certain milestones is really important. And this brings in the importance of that feasibility when you're looking for funding for research. So let's talk about research methodology. I'm sure you will have seen the pyramid of the hierarchy of evidence before. What this explains is the relative strength of evidence from experimental research. At the bottom, we have expert opinion or editorials, and at the top, we have synthesized evidence, things like systematic reviews and meta-analysis. However, some caveats. Synthesis of poor quality evidence doesn't make for a good quality systematic review. So your systematic review and meta-analysis are only as good as the trials which are included in those in those reviews. So it's always a bad idea to try to bring together lots of poor quality trials. You won't learn anything more. The other thing is randomized controlled trials, although really high on our hierarchy of evidence, level 1A evidence, they aren't the answer to every research question, and it's not practical or feasible to run an RCT for every question that we have in research. So what are the different types of trials? So let's talk RCTs. These are comparison studies. Participants are randomly allocated to a treatment or intervention and then compared to a control or a placebo group. And what we're looking to see is what's the effect of that intervention in this cohort of patients. The advantages is there's an unbiased distribution of confounders. What we mean by a confounder is a variable which we can't control, which may affect the results of your trial or your study. So having this randomization helps to reduce the risk of the confounding factors being biased. Randomization also reduces selection bias. So that is when researchers try to cherry pick cases that they think are more likely to respond to treatment and putting them in the treatment arm, for example. RCTs, we can usually have an element of blinding, whether that's the researchers, the participants, the analysts, and they're usually prospective, which allows you to have much more control over your data. The disadvantages are that they are expensive, they are very time consuming. Some of the big RCTs can take up to 10 years from conception to completion. There are ethical considerations. You need to check that what you're proposing is suitable and acceptable for patients. And they're very difficult to run in rare conditions or things that have rare outcomes. So a crossover trial. So this is when each participant has both therapies. So a patient may be randomized to start treatment A and then start treatment B. So the advantages of this type of study is that the participants can act as their own controls because you can compare how they responded to standard of care versus how they responded to the treatment. And the best control is the same person. The best control is you before you had the treatment. It removes patient variation when you're interpreting groups. It reduces the sample size you might need. And ethically, all patients do receive some treatment. Blinding, it may be possible to maintain blinding throughout the trial too. However, they're not usually used for treatments that have permanent effects, because once you've got the effect of one treatment, that may alter the responses to subsequent treatments. So you need to have adequate washout periods where the first treatment is washed out of the system and is no longer effective before you start the second treatment. Let's think about some observational studies, a cohort study. These can be prospective. So what we mean by prospective is the data is all collected and everything is looked at from the start of the study. or retrospective, where you go back and have a look at the cohorts based on existing data. So what we're trying to do is have a look at patients or participants who have been exposed to the outcome of interest, following them up over time and seeing what the effect of that exposure was. So it's really useful if you're trying to find out if a certain risk factor is associated with a condition or disease. And it also describes the natural history of the disease or the incidence of a disease. So it's ethical because we're not doing any interventions to patients. It can help you to demonstrate cause and effect. You can look at multiple exposures or variables. However, you do get a lot of loss to follow-up. If you are looking at a long latent period between your exposure and the incidence of disease, you may find that that is a difficult trial to run, that you lose participants, and that may introduce bias because the participants that may remain may be different from the ones that you lose from your study. You may find that you get missing data, and there may be unknown confounders that we're not able to control for, particularly if studies are running for a long time. Often you do need large numbers of participants, and you'll need a larger sample size if your outcome of interest is rare. So a case control study. These are usually retrospective, so going backwards and having a look at what the outcome has been. So what happens is you have a group of patients with a disease, and you go back in time and you say to them, were you exposed to these various factors, to see if you can determine which variable was associated with the development of the disease. You try and match a control group, and sometimes this can be difficult to define. But the advantages of this is that they're quite good for rare outcomes. They're quite cost efficient. And if there's a really long time between an exposure and the effect, a case control study can help you to reduce the amount of research time needed to wait for those effects to happen. So a case control study may be better than a cohort study if you have a long latent period. And we can look at a lot of variables. However, again, there's always going to be confounding that is unknown and that we're not able to control for. You may get sampling bias. So, for example, if you're looking at rare cases that you're identifying from a tertiary referral centre, you may find that these patients included in the study are systematically different from patients who did not access care in a tertiary centre that you weren't able to include in your trial. And you also get recall bias. Recall bias is where participants in the study remember things differently because they have the disease. So database studies, these are becoming much more common. There are big databases, national cancer registries, for example, and there's increased archiving of data, which means there's a lot of data that we can explore, and you can pick out your cohorts quite easily. So one advantage is because the data has usually been collected for a purpose outside of research, such as coding or financial billing, it reduces the observer or researcher bias. And the data is often standardized, and they're quite inexpensive studies to run. However, the data may be incomplete, and this can lead to sampling bias. And it might not be ideally suited for testing your proposed hypothesis. So you have to have a think about whether a database study is going to give you the right information that you need. So let's come on and think about synthesis of evidence for our secondary studies. So under this umbrella, you have your narrative or simple literature reviews. These are where you go and you research a topic, you summarize the evidence, and you present it in a research paper. A systematic review is when there's been a predefined literature search and a systematic overview of all of the included studies that have come up as a result of that literature search with a systematic data extraction, and I'm going to go through that in a bit more detail. Whilst meta-analysis are statistical synthesis of data from trials included in a systematic review. So how do you do a systematic review? First of all, you'll select your topic and do your research to design a research question, which again should be a PICO question. You then need to design a search strategy, and this is often done with a librarian to help you think about the right terms and how you're going to search these terms. You then want to specify which databases you're going to search. So common medical databases are things like PubMed, Embase. You'll want to, when you have your included studies, check all of the references to look for relevant work. And then once you've conducted your literature search, you're going to get lots of hits, sometimes thousands of hits, and you then need to look at every single abstract to determine whether it meets the criteria to be included in your systematic review. Once you have the included studies, you then need to do the data extraction from each included study. This should be done by 2 independent reviewers, and you'll need a third reviewer to solve any disagreement. So if there's any data which isn't matching between the 2 reviewers, a third reviewer will need to arbitrate that to decide what the final extraction of the data will be. All systematic reviews will have a quality assessment and a risk of bias assessment, so we know what quality the included studies are. And you might find that you need to go back and redo your search as the review progresses. And it's common right at the end of the review to rerun your literature search to try and find anything which has come up in the interim period between the initial search and the finalisation of your data. So meta-analysis will combine the results of individual studies using statistical methods. So commonly this will be presented as a forest plot. So what a forest plot does is it shows you the information on individual studies. So you can see in the example here, each horizontal line is an individual study. Now the black boxes represent the point estimate of that study. So in this case, it's the odds ratio. So Smith et al. Have showed that you're about a 1.3, you have a 1.3 odds ratio, so 1.3 chance of getting X disease, for example. Now the size of the black box represents the size of the study, and the horizontal lines represent the 95% confidence interval. Now, you'll notice that the bigger studies have a narrower confidence interval because you can be more sure of those results. You then have this diamond at the bottom, and the diamond is the combined estimate of all of these included studies. And the horizontal width of the diamond tells you the 95% confidence interval of the combined point estimate of the studies. You may find that you get the I-squared statistic. This is a statistic that tells you about the heterogeneity of the studies, so how consistent the studies are that have been included in the meta-analysis. So I encourage you to go away and have a look at some forest plots and think about how you might interpret them, as these are often quite common exam questions. But some thoughts to think about when you're thinking about doing a systematic review or meta-analysis. It's good when you want to combine existing good quality studies which have different results to try and get that overall estimate. They will increase the size of your study population, and they can allow you to do some subgroup analysis, and this can generate hypotheses for future studies. However, you need to think, is a meta-analysis on this topic sensible? So, did the studies included in the review address the same research question? And were the studies designed to address the research question which you are asking? Are the studies that are included of comparable quality? And are they actually comparable? So did they test the same population? Did they look at the same setting? Did they offer the same treatments? Or were there different study protocols, which may mean that you shouldn't be combining the studies? And in that sense, sometimes a narrative or a simple literature review is more powerful than trying to combine the results. Okay, so I'm going to spend the last 5 to 10 minutes just talking about research governance and ethics. So why is research governance important? So it improves the quality of research because it means that we have robust processes to ensure that research is being conducted in the proper way. And this protects the public and patients and means that the public and patients feel confident to participate in research studies. So research should maximise the benefit to individuals and minimise risk and harm. And we need to protect the dignity and the rights of those individuals involved in research. Participants always need to have made informed consent. So this would mean that they'd have had opportunity to review a valid participant information leaflet. They had had time to speak to a trained researcher. They've had time to go away, usually 24 to 48 hours, and think about whether they want to participate. And your information should be accessible to all groups, so you should make sure you can get your information in different languages, or you may need to have support if there's a disability in order to translate your information leaflet to an accessible format. And when consenting for a research trial, it should always be by trained researchers. There's formal consent training on how to consent for research, and it is different from consenting for a procedure. And you need to make sure that the patient is always, fully aware of all of the risks and benefits of the research study, and that they know that they can withdraw at any time and that won't impact on their clinical care. You also need to have clear accountability for the proper conduct of the trial. This usually falls with the PI, which is the principal investigator of the study, and they are responsible for making sure that all of the data is collected and stored in a safe way, that all researchers have been properly trained and are appropriate personnel to be involved in the research study. And there are both internal and external audits of research documents. So things like your consent forms will be checked, your site file, which should contain all of the information pertaining to a research study, should be well put together, thorough, and should have all the relevant information. And this will be audited during the course of your research to make sure your research is being done correctly. So to touch on ethics, there is a HRA decision tool. I've put the link at the top. If you're considering doing any form of research, you should check whether you need NHS ethics, and then you write a proposal and submit that to a research ethics committee. There are regional committees and they will have lay members on them, and they do a robust, proportionate review of your proposed project and will let you know whether they will give ethics approval or whether you need to make amendments to satisfy them that your project is being done with the appropriate ethics. Of note, if you decide after you start the study that you want to change the study in any way, you will need to go back to the REC committee for a substantial amendment to get those research protocol amendments approved. Otherwise, they can come in and shut your study down. So, career pathways. So, you can do academic training at any point in your medical career, but some opportunities to do that may be at medical school in the form of an intercalated BSc or MB PhD program. In the foundation program, you can apply to an academic foundation program where you'll be given specific allocated time for research. In your specialist training, you can do an academic clinical fellowship, which can either be integrated into your training or as an out-of-programme for research experience, where usually you will do a higher degree like an MD or PhD. And then following an award of a higher degree, you can apply to be a clinical lecturer. And this is usually done part-time alongside your clinical training. And that usually takes up to 4 years, so 50% clinical, 50% research. And then after CCT, you're then looking to progress your academic career to become a senior lecturer or a senior clinical fellow, usually with an academic position at a university. So there's lots of different ways to get involved. You don't have to have been involved from the start at medical school. You can enter into research in your specialist training. It's all about the sort of individual circumstances and what you would like to do. And I'm very happy to chat to people if they'd like to have a chat about careers in clinical research. So please get in touch with Mr. Abdul if you would like to have a chat with me, and I can provide you with my contact details, and I'm happy to take any questions via email as well. Thank you very much.